Pietro, a Ana Paula está aí perto de você, Pietro? Pietro? Ele está com o som cortado.
What do you think, Paulo? Is the YouTube connection working? Do you hear me as first?
You are mute, Professor Paulo.
Okay.
Paulo, você está com o microfone fechado.
Desculpa, é verdade. Desculpa, Valente. Ah, tá. Maybe I think that the YouTube link was closed unexpectedly, and he's trying to create another one.
Okay, let me know when you are ready.
Okay.
Professor Paulo.
Sim, Mauro.
Já fiz o novo link, já criei a transferência, né, do link anterior para o link atual.
Certo.
Já informando na descrição as intercorrências técnicas que a gente teve com a plataforma do YouTube, tá?
Então alguém que acessar agora o YouTube vai ter acesso já a esse momento que nós estamos?
Já estamos, como o senhor iniciou o streaming, já está rodando no YouTube também.
Ok, perfeito. Então nós vamos dar um início à videoconferência. Sylvia, it's an honor for us to have you here, to having accepted our invitation for this webinar. And Silvia Espriano is a professor of Politecnico de Torino. She's an excellent researcher on what David Williams called the bioactivity zone. So today she's going to talk about that. And thank you again, Sylvia. It's with you. Thank you. And thank you for the audience.
Thank you very much, Paolo, for your kind presentation, for organizing this webinar, and thank you all for attending it. The title of this webinar is Titanium Surfaces Modified for a Specific Host Response. As first, I would like to make a very brief overview of the state of the art about titanium surfaces for bone contact application, that means dental or orthopedic implants. In the past, the research was very much focused on osseointegration. And as a result, we have currently a variety of clinically tested surface treatments or coatings for osseointegration. But despite this, we have dental or orthopedic implant failure. In the case of the dental implants, they are often associated to peri-implantitis and epithelial downgrowth. So nowadays, the research is much more focused on multifunctionality. That means, beside osseointegration, that means a specific response and several actions together on the same surface. And I would like to give you an overview of our research in Politecnico about the integration of titanium with soft tissues such as gum, or some surfaces for a low risk of infection and anti-microfouling or antibacterial behavior, and something about the modulation of the inflammatory response. Okay, so the first topic, integration with soft tissues. In this case, concerning the dental implant, we are focusing on the collar that is in contact with the gum. And what is already known is that if we want to avoid the epithelial downgrowth, we have to avoid the inflammation of the connective tissue, and we need a very healthy adhesion and proliferation of fibroblasts, because there is a strong interplay between the two effects. And at the end of this cascade of events, we can have bone resorption and implant mobilization. The second goal concerning the collar of the dental implant is a sealing action against bacteria infiltration. The gum is the first barrier against bacteria. So we need a strong and healthy soft tissue adhesion to the implant in order to avoid bacterial infiltration. And you here in red you can see some researches we are working on. The first one is the guided growth of fibroblasts on the collar, or some antibacterial strategies applied to the collar, or the modulation of the inflammatory response. The first strategy, just using a physical approach and just modifying the surface topography and morphology. What we know is that fibroblasts are rugophobic, so they don't like high roughness. And there is a threshold that is about 0.3 microns, which must not be overcome in order to have a good fibroblast adhesion. And we know that on the other side, bacteria are rugophilic. They like very much valleys on the surface. And again, there is a threshold that is 0.2 microns in order to avoid biofilm formation. So at the end, we have a threshold of 0.2 microns that is a roughness able to have a good fibroblast adhesion, and it does not encourage bacteria adhesion. So the current state of the art is that usually the surface of the collar of the implants are very smooth in order to prevent the bacteria adhesion. But we know that a very smooth surface is able to promote a pro-inflammatory response of macrophages, so it is not actually the best. So we tried with a physical modification of the surface to get topography able to avoid the risk of infection and to favor the formation of healthy soft tissue. And through the collaboration with Dr. Varkomica, who is attending to this webinar at the Graz University, we performed surface texturing through E-beam and inturing also with laser. You can see here the electron beam. When it is applied to the titanium surface, you have the melting of a surface layer that is some tenths of microns thick, and then you have a rapid solidification of the material. And if you properly play with the parameters of the process, you can get some microgrooves. You can see them here in the SEM picture. And we selected to have microgrooves which are 10 or 30 micron wide. This is because this is almost the size of fibroblasts. And we were able to get a roughness that is around the threshold of 0.2 microns we just explained before. And the biological response of primary human fibroblasts to this surface is that if you have just a mechanical polished surface, you can see a pretty random distribution of fibroblasts. But on the E-beam structured surface, you can see a very nice alignment of fibroblasts through the contact guidance effect along the microgrooves. You can see here the degree of alignment of the fibroblast along the direction of the grooves. And you can see that when you have the grooves 10 microns wide, the alignment is very nice and the statistical dispersion is very low. On the other side, if the grooves are 30 microns wide, you have almost a random distribution as it is on the mechanical polished surface. And this is also the case of very small 5-micron grooves, because in this case, we were not able to get such nice grooves on the surface. They are too small in dimension, and the E-beam technology is not able to get nice grooves. And so the cells are not guided by these grooves. And it is well known that the contact guidance effect is due to an adaptation of the cytoskeleton spread. This is very well expressed on the 10 micron wide grooves, because the fibroblasts are able to touch both the side walls and the bottom of the microgroove because of the dimension. So they are very well guided along the grooves. And with this effect, we have a more stable gum sealing and a lower risk of the periodontal pocket formation, and so at the end, lower epithelial downgrowth and bacterial infection. This is already known in the literature. What we explored as a new outcome is to obtain this effect by new techniques such as E-beam and laser texturing. But the E-beam surface texturing has also several other effects. One effect is on the microstructure. Because of the rapid cooling, fast cooling of the melted alloy, you get this high density of lamellae and grain boundaries. And if you look at the nanotopography of this surface, you can see very sharp nano steps on the surface. This is a secondary effect of the E-beam surface treatment. And as last, you can also a different crystalline structure, because you have the formation of martensite, which is an orthorhombic crystalline structure of titanium. And if you look at the formation of biofilm by Staphylococcus aureus on this surface, you can see that at 1 day and 2 days, you have a lower adhesion and development of biofilm on the surface. And it is very interesting to look at this graph on the right, because you can see that you have this effect on the 10 wide microgrooves, on the 30 microns wide microgrooves. And and also when you very smoothly polish the surface, so you remove the micro grooves, that is the P10 and the P30 surfaces. In any case, you have the antibacterial effect. So it is not strictly related to the microgrooves. And you can see that if you perform a high thermal treatment, that is the EB10HT, again, you have the anti-adhesion effect, but in this case, you have no martensite. So it is not related to the martensite. And at the end, we can say that the mechanism of anti-microfouling action of the E-beam textured surface is due to the high density of nanosteps on the surface. And this can be explained because bacteria are very smaller respect to fibroblasts, and they have a stiff membrane, and they try to have a high stability of the and shape maintenance. So at the end, they are not able to maintain their shape on this surface, so you have a reduced adhesion on nanotextured surfaces. And this is a new outcome coming out from our research, because it was not reported in the literature before, and it can be get both on pure titanium with that is an alpha titanium, on titanium 6,4 that is an alpha beta titanium alloy, or on a beta titanium alloy such as the titanium molybdenum 1. And this is a passive strategy. So you are able to avoid the colonization of the surface by the bacteria, even if you are not able actually to kill the bacteria in the surrounding. But you can imagine to couple these passive strategies to other strategies with an active antibacterial action for a synergic action. And what is also interesting is that if you use laser instead of E-beam to get the same time of texturing, you can observe the same effect on fibroblasts, that is the contact guidance effect, and the effect of bacteria is slightly different. So you have not a lower adhesion, but you have a very low proliferation of the biofilm. And this can be explained because the nanotopography of the surface, you can see here in the picture on the right, there is a nanotopography actually, but it is much more smooth than it was on the E-beam treated surfaces. And it is reported in the literature. Then when you have a nanotopography, you can have a reduction of the area, that is the blue area here in the draft, that is available for the proliferation of the bacteria within the biofilm. So probably we are in this condition on the right of the draft, and the topography has the right features to reduce very much the area of the bacteria that is suitable for the proliferation of the biofilm, which occurs only in some directions on the top of the first layer of bacteria. So we want to go further in this research. I think that it is very interesting in order to understand the mechanisms of the biofilm formation. Okay, this is the end of the first story. The second story is about a different modification through a chemical approach, not a physical one, as it was the first. And we tried the addition of keratin, and also we doped keratin with silver ions. What is already known from the literature here in blue is that keratin is able to enhance able to be an active antibacterial.
Silvia? Vamos aguardar pouquinho. Vou falar com ela aqui. Silvia, volta para que. Please back to the effects of keratin on fibroblasts, because the internet was down for a few seconds.
I'm really sorry. Now you are hearing me?
Yeah, with keratin and fibroblasts.
Oh, sorry. So only the keratin, right?
Yeah.
Okay.
When you were talking about keratin and fibroblasts.
Okay. So it is already known in literature that keratin is able to enhance the fibroblast adhesion and proliferation. And we know that silver has a strong antibacterial action through different mechanisms. But we also know that silver is cytotoxic. So it is not easy to find the proper dose of silver able to have an active antibacterial action without cytotoxicity. What we explored at Politecnico is to couple keratin to a titanium surface and to dope it with silver in order to get an active antibacterial action and an enhancement of fibroblast adhesion. And we used keratin in different forms, random nanofibers or aligned nanofibers, or single keratin grafted molecules on the surface, or a continuous coating of keratin. And we obtain similar results. I will show you some results right now. And an interesting point is that keratin can be extracted from the waste of the wool and meat production, so with a circular economy approach. You can see here some nanofibers of keratin with a random distribution on the left and with an oriented alignment on the right. And you can see that after 28 days of soaking in water, here on the right, the nanofibers are almost unchanged, so they are very stable. And we can enhance the mechanical adhesion of the nanofibers to the titanium through plasma activation. And you can see that keratin can also be deposited as a continuous coating here on the right, or just through functionalization with some single molecules grafted on the surface. You cannot see here by your eyes the molecules, but if you perform an XPS analysis, you can see the amino groups of the amino acids of keratin and also the presence of sulfur that is typical of some amino acids in keratin. And which is the biological response? You can see that human gingival fibroblasts have a very good adhesion on the surface without any cytotoxicity. And if you compare in the middle of the slide, if you compare the polished and the polished plus keratin coating, you can see that the number at one day, the number of fibroblasts is quite higher than on the polished surface. And if you have aligned nanofibers of keratin, as it is on the right, you have again a contact guidance effect on the fibroblasts as the microgrooves we have shown before. So what is interesting is that fibroblasts are crucial for the inflammatory cascade, and so an easy implant colonization can be useful for preventing a pro-inflammatory cascade, and we want to verify this. And as I told you, you can have also a contact guidance effect if you are able to align the keratin nanofiber. And as a second step, you can dope with silver the keratin fibers or coating, and you can see again the XPS signal. On the left, you have just keratin, but on the right, you have the modification of the silver peak in XPS profile of when you add silver. And you can see that there is a shift of this second peak and that there is a change in the intensity. This is related to the presence of silver that is chemically bounded to the sulfur. And if you look at the silver peak in the XPS analysis, you see just the same effect. These peaks can be correlated to the presence of silver as chemically bonded to the sulfur in keratin. And what is very interesting is that if you put this surface with keratin and silver in water, you get no release of silver ions in water. Sorry, this means that you have a strong chemical bonding of silver. If you put this surface soaking in broth for cell or bacterial culture, you have the release of silver at 1 day with this medium for cells, and the maximum is at 2 days if you use the bacterial broth. But the silver is not free. It is strongly bounded to the proteins inside the medium, or it is within salts, because you have also chloride ions, and silver chloride is very stable. And if you look at the behavior of the formation of Staphylococcus aureus biofilm, you can see that by increasing the amount of silver, you have a larger antibacterial effect. You can see also on the right, in blue, you have just the polished surface with keratin, and then you have 3 different amounts of silver with increasing antibacterial activity. And you can see also on the left, on the top, you have the titanium polished with keratin with a lot of alive bacteria. And on the other side, when you add silver, you can see a lot of dead bacteria. So in this case, it is not a passive, it is an active approach able to kill bacteria. But what is very interesting is that if you put human gingival fibroblasts on the same surfaces, they are biocompatible, and you have not any evident reduction of the amount, the number of fibroblasts. So you can get by this doping of keratin a balance between an antibacterial action and biocompatibility. That is not trivial. We have a lot of antibacterial elements or compounds or biomolecules, but it is not easy when you put these biomolecules on a surface to find a right balance between biocompatibility and antibacterial action. So we want to go further in this research. Here you have some papers about this topic and about the microgrooves. Another story, another research at Polito. If we leave the collar and we look at the bone, at the implant in contact with the bone, we developed some modification of titanium in order to have osteoconductive and osteoinductive ability, and again to have an antibacterial action, and also an osteoinductive ability through functionalization with some biomolecules such as polyphenols. The first results concerns the osteoconductive ability through a micron and nano topography. In this case, the nano topography was obtained through a chemical etching, and it is interesting because it is a multi-scale topography. You have such roughness of about 0.3 microns, and then you have a nano roughness overimposed to the micro one, just of 10-20 nanometers. And this is because during the chemical etching, you have the formation of a titanium oxide layer that is about 400 microns thick, and you can recognize the color of the titanium oxide. You can see here in this small figure in the bottom right. And it is about pink and green, that is the color of this thickness. And you have such a typical topography, but you have also a peculiar chemistry, because the surface is very rich in hydroxyl groups. You can see again the XPS analysis. This is the titanium peak, and you can recognize two peaks. One is due to the titanium oxide, and the other one in red is due to the hydroxylation and hydroxide groups on the titanium oxide. And by zeta potential measurements, you can see here that there is a great difference between polished titanium, that is the curve in gray, and the treated titanium, that is in green. And you can see an evident shift of the isoelectric point. And in the case of the treated surface, the isoelectric point is around pH 2, so in the acidic range, because these hydroxyl groups have a very strong acidic behavior. That means that they are easily deprotonated. If the liquid in contact with the surface has a pH of about 4.5 or higher, all of these OH groups are completely deprotonated. This means that at physiological pH, here at around 7, you have a surface with a deprotonated hydroxyl group. And we applied this chemical treatment to dental implant, to an acetabular cap obtained through additive manufacturing, and also to a porous laryngeal implant with microbeads, obtaining the same topography and the same chemistry. And if you characterize the surface through contact angle measurements in order to measure the wettability of the surface, you can see that after the treatment, the wettability is higher. You can see also on the right a dental screw, and you can see this is a drop of water, and you can see that the water is attracted by the surface, and it is able to work on the surface and to complete wet the surface. This does not occur usually on a blasted or just a machined surface. And if you put this surface in contact with SBF, that is an inorganic solution with the same chemical composition in terms of salts of the biological fluids, you can see the precipitation of hydroxyapatite, because the presence of the deprotonated hydroxyl group is able to attract calcium from the simulated biological fluid, and then phosphate, and to get the precipitation of hydroxyapatite, that is the mineral component of bone. And the behavior with osteoblasts of this surface, you can see that at one day you have a larger amount of osteoblasts, and you have a strong differentiation of the osteoblasts. You can see that they are very flat on a usually etched, such as SLA, dental surface, while they are 3D on the treated surface, because the topography is able to induce this differentiation. And we tested also fibroblasts, and you can see here on the bits of the laryngeal implant that without any treatment, the number of fibroblasts is low, but on the treated surface, the proliferation of fibroblasts is higher. So in this case, probably the roughness is enough low to have a good behavior both of osteoblasts and fibroblasts. On the other side, if you put macrophages on this surface, you get a very low number of macrophages. You can see here the graphs about the metabolic activity and also the expression of different cytokines by the macrophages. And you can see that at 14 days or also at 10 days, you have a significant statistical difference. So this surface is able also to have an action in controlling the inflammation with an anti-inflammatory behavior. And as last, again, we have a nanotopography. So again, we have passive strategies against bacterial adhesion and biofilm formation. Some papers about this surface. And we can go further. We can dope this nanotextured surface with silver. In this case, we get silver nanoparticles. And it is very interesting because you have very low, very small nanoparticles, lower than 10 nanometer in diameter. And this is a section in the middle of the slide. You have a section of the treated surface, and you can see the titanium oxide going down the surface along the grain boundaries of titanium. And you can see that some bigger nanoparticles are formed below the surface, but they are very small on the external surface. And the XPS again tell us that silver is present as a metal in this case. And we tested different concentration of silver nitride, that is the precursor for the formation of the silver nanoparticle that is added during the etching of the surface. And by using different concentration of silver nitrate, you get a different amount of silver on the surface. And you can see in the bottom right the release of silver ions in water. And in this case, it is very different from what we observed with keratin. In this case, we have a release of silver that is lower in the case of the lower amount of silver on the surface, that is the yellow curve, and it is larger, the red curve, when you have a larger amount of silver on the surface. If you look at the behavior of the bacteria, again, biofilm formation by Staphylococcus aureus on this surface, and if you look to the orange curve, you can see that with a high amount of silver, you have, the blue curve with a high amount of silver, you have an active antibacterial effect on the planktonic bacteria at 24 hours or some days. So this means that the silver released by this surface is able to kill bacteria in the surrounding, not only on the surface of the implant. And on the right, you see the effect on the biofilm. So on the surface, on the bacteria developing on the surface of the implant. And you can see again in blue that after 1 day, the bacteria are completely killed on the surface. So a very high bactericide action with a high amount of silver. On the other side, if you look at the viability of age FOB mesenchymal cells, we see that the curve in blue, we have a strong reduction of cells. So this surface is bactericide, but it is also cytotoxic. If you reduce the amount of silver, like in the orange curves, you can see that you have no effect on the planktonic cells, so you are not able to kill the bacteria in the surrounding. You are able to kill the biofilm on the surface. The curve is almost the same that it was for the higher amount of silver. And in this case, the biocompatibility is good. So when you use nanoparticles on a surface, it is not easy to find a proper balance between a bactericide or a bacteriostatic behavior and cytocompatibility. With a low amount of silver, you can try to do this, but as you can see, the biocompatibility is not perfect. So with keratin, I think that the result is better. So it is much easier to find a balance between the two properties. With the nanoparticles, it is much more difficult. Some papers about this. And then the last story for you, you can add polyphenols to the nanotextured titanium surface. We tried with different polyphenols in a first attempt with polyphenols extracted from green tea. So the process is almost the same. But after the chemical etching, you soak your surface in a solution containing polyphenols. And you have, in this case, to add also calcium inside the solution, because you can see this mechanism on the bottom right of your slide. You can link the polyphenols to the surface through the action of calcium, because you have a negative charge on the titanium surface, you have a negative charge on the polyphenols, and you have a positive charge on the calcium ions, so you can use these ions as a linker between the surface and the polyphenols. And this is the chemical and physical characterization of the surface. If you make just the XPS analysis, you can see that the amount of carbon after functionalization with polyphenols is larger. So you have carbon that is the main element of the polyphenols. You have a lower amount of oxygen because you are covering the titanium oxide. And you can see that the XPS analysis is not able to detect titanium because of the thick layer of polyphenols on the surface. And you can see also the presence of calcium because of the mechanisms I described to you. And you can also see with this on the left an enlargement of the profile fitting of oxygen. And without polyphenols, you can see the titanium oxide and the OH groups. You already know this picture. But if you add the polyphenols, the profile fitting is completely different because of the presence, and this is a confirmation of the presence of the polyphenols. And also the wettability is different, with a larger wettability, a lower contact angle when you add the polyphenols. And if you perform the Folin and Chocalteau test, that is a chemical test evidencing the activity of the redox activity of a biomolecule. You can see here that you have a chemical reduction of the reagent, of the Folin-Chocalteau reagent. You can detect this because when you have the reduction of the reagent, you have a change in the color of the solution in contact with the surface. And so the polyphenols grafted on the surface are able to have a chemical reduction action that is usually connected to an anti-inflammatory action. So this is very interesting. And these polyphenols are fluorescent, so you can see by microscopy the presence of the polyphenols on the surface as aggregates. And with a collaboration with the Kyoto Institute of Technology, we explored the biological response of this surface, functionalized surface, with respect to CUSA cells. And what is much more interesting in this research is the application of FTIR to the cultured cells on the surface. This is not so usual, but it is very interesting because you can detect the presence of hydroxyapatite. You can see here in the blue area, some peaks, the strongest peaks are due to the presence of hydroxyapatite. And you can also detect the signals due to the presence of proteins, lipids, and organic components in the red box. So you can compare here just the treated surface without polyphenols and the surface with polyphenols. And you can observe an evident increase of both the peaks in the blue and red areas. So you have the ability of these mesenchymal stem cells to be differentiated on the functionalized surface and to become osteoblasts and to produce hydroxyapatite. Also, the presence of osteocalcin was evidenced, and this is another result highlighting the differentiation of the mesenchymal stem cells. And also Raman spectroscopy can be applied to the cultured cells. And again, you can see that the peaks related to hydroxyapatite are much more evident when you put the cells in contact with the functionalized surface. So at the end, we can say that the chemical treated titanium alloy surface itself is able to induce osteoblast differentiation, but polyphenols are able to enhance the deposition of mineralized and non-mineralized extracellular matrix. And I want to mention a very interesting European project we had in collaboration with Professor Tambasco de Oliveira, and the project is called Nut4More. In this project, we explore again polyphenols coming from a different source, that is grape pomace, so a waste of the production of wine, both red and white grapes. And you can see here in the bottom, we explored a wide range of surfaces, polyphenols grafted again using calcium as a linker on a titanium treated surface with a similar approach to the green tea polyphenols. But we explored also another chemical treated titanium surface, which has a nanotexturing, but not the deprotonated hydroxyl groups. So in this case, we get the functionalization by using amino acids, and there is a chemical affinity between amino acids and polyphenols. So you can get on the surface a layer of polyphenols entrapped, embedded inside a layer of amino acids. Or we used a chitosan coating on the titanium surface, and we enriched the chitosan coating with polyphenols. And we grafted polyphenols also onto bioactive glasses. In this case, you do not need calcium as a linker, because the bioactive glass in contact with a fluid is able to form a silica gel on the surface, and polyphenols are embedded within this silica gel layer. Or you can also graft polyphenols to hydroxyapatite. In this case, calcium is present in hydroxyapatite, so it can be used as a linker for grafting polyphenols. The biological results are still ongoing, so I cannot describe them right now, but it is very interesting, and I want to thank Professor Tambasco for its great support and cooperation in this project. Some papers, and now the conclusions. The osseointegration requirements of dental implants are almost achieved, but the new goal for implant titanium surfaces is a multifunctional action. That means as first osseointegration ability and then low risk of infection and immunomodulation. In the case of transmucosal implants, a specific goal is a strong interaction with soft tissues as a barrier against bacterial infiltration and epithelium downgrowth. And we can get this by oriented grooves through E-beam or laser surface structuring, with a good integration with soft tissues through a contact guidance effect, and an anti-microfouling, so a passive action against bacteria. With the addition of keratin to the surface, we can enhance fibroblast proliferation, and keratin can be doped with silver for an active antibacterial action, so a balance between cytocompatibility and an antibacterial action can be found. The micro and nanotopography of titanium surfaces can enhance tissue integration with an anti-microfouling passive action and control of inflammation. You can get this by several techniques, such as the E-beam or also chemical etching. And you can try to dope the titanium surface with silver nanoparticles in order to induce a bacteriostatic or a bactericide ability, depending on the dose of silver. But in this case, it is difficult to get a balance with cytocompatibility. And then you can use surface functionalization with polyphenols from green tea or grape pomace in order to enhance the deposition of mineralized and non-mineralized extracellular with a large osteoinductive effect. So thank you very much for your attention. I want to thank all the institute which are collaborating with us on this research, and all my GLANS research group.
Is that Po? The river?
Yes, that's all.
Thank you.
It was a bit too long. I'm sorry.
Okay, excellent. Thank you very much. Thank you very much indeed. Maybe we can open. You stop the presentation. Oh, you keep there, right? You keep the presentation.
Okay, so if you need, I can go to some slides.
Yeah. Any questions?
Hi.
Hi.
Actually, I have a question. Nice to meet you, Sylvia. Thank you for the opportunity to be here and watching our presentation. Thank you very much for your presentation. Actually, I have two questions. First one is at the beginning of your presentation, when you mentioned that you use plasma to functionalize the titanium. And I'm just wondering about the reasons that you use plasma to functionalize the titanium. I mean, is it because to reduce the hydrophobicity of the surface before you developing the coating, or?
Yes, it is because by plasma you are able to remove all the organic contamination on titanium, because usually on titanium, just because of the exposition to the air, you have a lot of organic contaminants. And you can also induce the formation of some hydroxyl groups. And in this case, these hydroxyl groups have a basic chemical reactivity. So they, in contact with a fluid, they are able to get a positive charge. So they are protonated. And because of the presence of these groups, the adhesion of the keratin fibers is higher. The mechanical adhesion, I mean, of the keratin fibers is higher.
I see. Yeah, I super understand, because I have used plasma as well to functionalize the titanium surface. I was just curious to make sure that the reason was similar, or there were another reason to do this. Thank you very much for your response. I don't know if I can make another question or...
Yes, for sure. Welcome.
Okay. The other one is about the silver. It was not clear to me. I mean, you expect that about the action mechanism of the silver in this case, you expect the contact, the antimicrobial appropriate by contact, or the release, and once it's to the silver release, you expect that what is the concentration that you expect to release it from the system to make sure that you get the antimicrobial property? I'm sorry, a lot of questions. Yeah, it's about the silver. Yeah.
Yes. This is a very interesting point for me because I think that the action of silver in the case of keratin is related to the contact between the bacteria and the silver that is still bonded to the surface. Because you can see that if you put this surface in water, you have no release. So this means that silver is bonded within a covalent bonding with sulfur. Only some proteins are able to remove silver from the surface, but probably this silver is not biologically active because it is entrapped and bonded to the proteins. Otherwise, you cannot explain the biocompatibility. This on the, I don't know if you look at my mouse.
Yeah.
Oh, yeah. Okay. So it's much more clear. You can see here that even if the amount of silver is high on the surface and the bacterial action is really evident, the surface is biocompatible. This means that you have not the release of silver that is biologically active on the cells. And if you have a release of silver, it is entrapped within the protein. On the other side, the silver that is active on the bacteria is the silver bonded to the keratin.
Yes. And how about the concentration of silver? I mean, how do you know?
Yes, I don't know if it was, sorry, I didn't...
Sorry if I escaped from me the information.
I didn't report it in this slide. Oh. I'm sorry. I think that is below 1%.
Okay.
You can check it in the literature. I'm sorry, I have not the figure.
No. I was just curious about the action mechanism of the silver, because you incorporated the silver at the same time. We know that the silver can be released from the system, and and at the same time is biocompatible.
Yes,
I was just wondering about all of this information. Yeah.
Yes.
Thank you very much.
We want to go further in this research because I think that we have to understand a lot of things,
yeah,
but it is very interesting.
It's very interesting. Thank you very much for your answer and for sharing all this information.
Thank you, Erica. Now, Valentin.
So thank you, Professor Silvia, for this very great presentation and for providing us this lovely topic. I love the surface modification for implant materials. So I have two questions. One is more specific and another is more general. The first one is about the laser texturing that you mentioned that you were able to change the microstructure of the surface. My question is, if you change like the temperature, the time of the treatment, and so on other parameters, are you able to create a titanium dioxide crystalline structure like an Atenciosamente rutile, thinking about photocatalytic action and so on, to have a more antimicrobial action?
Well, I think that the e-beam technology is under vacuum. So there is not actually the formation of a thick layer of oxide. And I think that Professor Fernando, who is attending the webinar, can eventually explain you better the technology. With the laser, we are working in air. So in this case, we get also an oxide layer, and the color of the sample is different. So for sure, we have an oxide layer. We are just at the beginning in this research. So it could be that this oxide layer has different properties.
Oh, thank you. So yes, my last question is more general. We have seen so many studies in the literature dealing with antimicrobial surface, and many of them, they have a very strong antimicrobial effect in vitro, and also some of them, they have in vivo effect using some animal models. But we know that none of the surfaces have reached the market in terms of commercialization and so on. We have tried to write down, conducted by Professor Erica, trying to explain why the company, they do not have this type of implant yet. But what you face is that like many in vivo models, they do not resemble what occurs clinically in terms of pet implants, in terms of disease progression. They do not have the real bacteria or the complex bacteria that are in the pet implantitis and so on. So what do you think, what is the future of the antimicrobial surface? Because many of the surface, they have a burst release, and we know that periimplantitis is a chronic disease, and we need the effect in long term, but we have just in short term. Of course, we have many studies in the literature that are trying to increase this release effect, but still they fail. So we also have seen some smart surf that we can control the release by changes in temperature, changes in oxygen level, changes in pH, and so on. But I think we do not reach there. So the race, it's still like we cannot solve this puzzle. Like, so what do you think is our future?
Well, I perfectly agree with you. I can just add a couple of comments. As first, I think that it's very difficult to predict the clinical behavior of the surface. I agree that when you are in vitro, it is working well, also in some animals, small animal models, but when you go to the clinical approach, it's different. And nobody, usually, nobody says who is the clinical results. So at the end, you don't see on the market the products, and probably it is because the clinical results was not so fine, but it is not published. On the other side, I think also that the CE marking or the FDA marking is becoming so difficult when you add an active biomolecular compound on the surface, that it is very expensive and very, the risk is very high when you go through this market. So marking of these surfaces is difficult. And that's why we are working also just with some physical methods and with a very low threshold of ambition, I mean, just the passive antimicrofouling. Because in some way it is much closer to the market than some very strong antibacterial action that is very difficult to be put on the market. And as last, I think that I want to go through the keratin because as I explained right now, the action of keratin with silver seems at this stage not to be related to the release of silver, but to the contact between the silver covalently bounded to the surface and the bacteria. So in this case, we can thinking about a very long time of the action of the surface, because it is not released, but it is all to be demonstrated at this stage.
Oh, thank you very much. Very clear.
Thank you, Valentin. Now, Professor Grandini.
Okay, thank you, Paulo. Thank you, Silvia, for this exciting presentation. We worked some years ago in the surface modification with nanotubes of the titanium dioxide. And in this moment, we are working incorporating the silver and zinc nanoparticles in nanotubes on the surface of titanium alloys. What do you think about this?
Well, it's very interesting for sure. Our experience with nanoparticles on titanium, sorry, I don't know which is the slide, is not so nice at the end, because it is really difficult to get a balance between cytocompatibility and antibacterial action. Because if you have, when you have the contact of the cells with the nanoparticles, the silver nanoparticles, you have a cytotoxicity effect. It's very difficult to get the right balance, at least on pure titanium. Perhaps nanotubes can be useful to find this balance, but just on the titanium surface, working with nanoparticles is very difficult.
Okay, thank you.
Thank you, Professor Grandini. Martina Bertolini.
You are muted, Martina.
I'm muted, sorry. Thank you so much for sharing your results. Very amazing lecture. I have two questions in two different areas. So first of all, the part that you mentioned about the microgrooves for the fibroblasts. I thought that was very interesting because we currently do have in the market, at least here in the US, an implant with this technology. I don't know if you know, it's the BioHorizons company, and they call it the laser lock, and supposedly is made exactly for this purpose, to create like a good contact with the fibroblasts and the sealing. So I think you guys use like different groove sizes related to the size in width and in depth. So I wonder if you have compared your results with this that is currently available in the market.
Yes, thank you very much for this question. We started from the laser lock surface. We investigated it, yes. And then we tried to do microgrooves with a lower depth in order to be sure to avoid the bacterial contamination. But actually, we did not perform a comparison during the biological tests between the two surfaces. So we just investigated first the laser lock, and then we decided to move to a surface with microgrooves smaller in depth. And we obtained good results, but the comparison could be very, very interesting.
Yeah, I wondered if you guys were able to achieve like a better outcome than what they have in the market. So I was curious. Thank you for that. And then I think in a follow-up of what everybody's already asking about your coatings, both with silver and the polyphenols, I was wondering, so you mentioned about the water immersion, but because this is exposed to the oral cavity, we have like abrasive forces, we have other things that could play a role into removing or destabilizing this coating. So I was just wondering if you have investigated how stable it is or how good this connection is within the surface, because we obviously don't want this to be removed over time, right?
Yes, yes. Concerning just the nanotopography, so this surface, we performed a sort of simulation of the implantation procedure by using a bovine bone and by using just the same equipment for a human usual implantation. And we observed that the nanotopography is resistant to the friction applied during the implantation. When